So first of all, I'd like to join all of you in thanking the organisers, Pro-Immune, for bringing us all together for this fascinating and important meeting. Now, I'd just like to show this first slide. As a disclaimer, I'm the founder and CSO of Epitope, and Epitope designs and tests peptides for antigen-specific immunotherapy. And furthermore, I'll be talking a little bit about MS1467, which is one of our currently developing treatments for multiple sclerosis. So moving on, really I have three messages that I'd like to get across. The first is really what we understand effective antigen-specific immunotherapy to be. The second relates to the work that we've been doing on the mechanism of antigen-specific immunotherapy. And then towards the end, I'd like to give you some examples of the approaches that we've been taking in various different types of hypersensitivity conditions. Primarily, we focus on autoimmune diseases, but as you'll see, we've been doing some work in haemophilia as well. So, antigen-specific immunotherapy is, of course, a well-practiced approach in the field of allergy. And this slide just reminds us that, in fact, antigen-specific immunotherapy has been around for over a century. Freeman and Moon in 1911 published an account of experiments designed to induce antigen-specific tolerance by injecting increasing doses of a crude extract of the pollen they thought was causing hay fever in this individual. And their description of the results were very, very clear, although somewhat beautifully archaic. Clearly, if we could achieve what they were describing here for treatment of an autoimmune disease, we'd actually be very, very happy. So what I really want to talk about is the nature of the so-called or apparent protective immunity that's being observed here. In the case of allergy, I think intact antigen is given for immunotherapy. And there may be some logic to that, because in fact probably what's happening in allergy is a shift away from IgE production towards blocking IgG antibodies. So arguably there is some advantage to giving intact antigen. However, this is still quite dangerous because, of course, intact antigen will cross-link surface-bound IgE and stands the risk of inducing anaphylaxis. Attempts to treat autoimmune diseases with intact antigen have also faced complications. For example, an attempt to induce peripheral tolerance by administration of myelin oligodendrocyte glycoprotein, MOG, in the primate autoimmune encephalomyelitis model of multiple sclerosis actually exacerbated disease by inducing pathogenic antibodies. So administration of intact antigen in autoimmunity would be guarded against in this case. Furthermore, in models of type 1 diabetes, administration of intact antigen can induce pathogenic cytotoxic T-cells. So a number of years ago, we reasoned that it may be safe and more effective in autoimmune disease to administer CD4 T-cell epitopes. And over the years, frankly, this has proved quite successful. So here is a list of different models of autoimmunity that have been treated effectively by administration of T-cell epitopes from antigens associated with those diseases. And I'm highlighting a few of these studies here because these studies demonstrate a very important phenomenon. That is that single peptide epitopes can induce something called bystander suppression, a term coined here in Boston by Howard Weiner following his studies in oral tolerance. So what bystander suppression means in terms of peptide immunotherapy is that you can administer one epitope from antigen A, okay, and it will suppress the T cells specific for different antigens B, C, D, E, etc. within the same tissue, okay? So that is what the term bystander suppression means, and it was demonstrated in my lab by Steve Anderson, who demonstrated that administration of an epitope from proteolipid protein would suppress disease induced by epitopes from myelin basic protein, for example. Furthermore, there was evidence from diabetes where administration of any one of a number of different peptides would suppress disease involving many different antigens. So the first question that we really came across almost out of frustration was what you have to do to design peptides that work. And this arose from the observation that certain peptides would induce tolerance and certain other peptides wouldn't. And Steve Anderson again focused on those that didn't, surprisingly. I mean, most people would focus on those that did, but Steve focused on those that didn't because he wanted to understand why not. Okay? And this proved to be very, very important. He showed, as we all know, class 2 MHC is a relatively open structure, and peptides can bind to class 2 MHC in different registers across the MHC molecule. And actually what Steve was able to show was that a peptide that didn't induce tolerance actually bound back to the MHC in the wrong conformation because it was different to the way the peptide bound to the MHC after natural antigen processing. Okay? This is really important. It tells you that antigen processing restricts the way the peptides bind to MHC proteins, right? But when you add peptides back from the outside, they can bind in different conformations. And this was shown by Steve, but also by Emil Unanue studying the lysozyme model peptides. So we've designed, or we've created a design methodology whereby we produce peptides that are independent of antigen processing, and these are called antigen processing independent epitopes, or epitopes. So we've seen that peptide immunotherapy can work. We now know how to design the peptides. The next question is, how does it work? Well, there is no doubt that peptide administration can induce antigen or activation-induced cell death. However, this fails to explain what is an active mechanism of bystander suppression. Secondly, we could be inducing anergy. The question would be whether anergic cells can mediate bystander suppression. Alternatively, we could be inducing immune modulation, where we're now generating cells that are what Ethan was talking about yesterday, suppressor cells, so cells actively suppressing the immune response. And to cut a long story short, about 5 years ago now, we described a logical mechanism by which this works. And effectively what was shown was that administration of soluble peptides would kid T-cells into thinking that they were Th1 cells. But in fact, they're not fully functional Th1 cells. Repeated antigen encounter drives these cells through to switching off any inflammatory cytokines they were making and now secreting IL-10. The IL-10 then feeds back on antigen-presenting cells and blocks the ability of this antigen-presenting cell to present not only this epitope, but other epitopes as well. And this suppression is mediated by downregulation of co-stimulatory molecules, but also blocking secretion of cytokines like IL-12 required for new generation of an inflammatory phenotype. So I think this would explain if this dendritic cell here was presenting a number of different T-cell epitopes. This mechanism would explain how bystander suppression works. And certainly we've demonstrated that these cells here in transfer models are capable of bystander suppression. Now, importantly, this phenomenon of generation of IL-10, if you like, as a negative feedback process, has also been recognized not only in Th1 responses, but also in Th2 and even in Th17 cells. So I believe that cells induced by chronic antigen exposure can use IL-10 as a negative feedback process. And certainly in chronic infections, you see upregulation of cells like this designed to limit immune pathology of the immune response attacking the infection. In chronic allergic responses and fungal infections, you can see the generation of these cells. And there is evidence that IL-10 is driven up by chronic exposure to antigen in allergy as well. So here are examples of the infectious diseases that I was talking about just now, where in chronic infection with toxoplasma, leishmania, etc., you can see the generation of these IL-10 producing FOXP3 negative cells. And I mean, this is what we have observed. We've found that, in fact, the type of FOXP3 cells that Ethan was talking about yesterday are not essential for the generation of cells with this phenotype. However, that was in a very, if you like, artificial system where those cells are not generated in the repertoire. And I do believe that, in fact, FOXP3 positive cells probably contribute to the generation of this negative feedback mechanism. However, we have enough examples in chronic infections where these cells appear. I also mentioned allergy. And this is a very, very fine example of exactly what I'm talking about. This is some work from Chesme Actis's group in Davos in Switzerland. And what Chesme did was to follow a group of beekeepers throughout the season. And you'll notice what they did was to measure the proliferative response of PBMCs from these individuals to the phospholipase A, the allergen of bee venom. And they noted that after the beginning of the season, the response of the beekeepers' PBMC fell and remained low until the bees went to bed. Okay? And this correlated with exactly the type of cytokine shift that I was talking about just now, where the response in terms of IL-4 production, gamma interferon, goes down, but the response in terms of IL-10 production goes up. So you're seeing these people have memory cells specific for the allergen, and their cytokine production is shifting on chronic exposure to the allergen. So the question is, can we develop this approach to switch off the cells involved in autoimmune disease? Questions we've been addressing over the years are the optimal route of administration, the optimal dose of peptide, and more specifically, the mechanism involved. Now this is some experiments published from our lab at the end of last year, where we've taken the TG4 model. This is a T-cell receptor transgenic mouse, and this mouse can develop autoimmune encephalomyelitis spontaneously or following immunization. And what you see is if you give repeated doses of peptide subcutaneously, the higher the dose of peptide you give, the more the immune response is suppressed in terms of cell proliferation, in terms of IL-2 production, and in terms of gamma interferon production. Cells from this mouse spontaneously produce gamma interferon in response to antigen in vitro. On the other hand, as you increase the dose of antigen you give, the levels of IL-10 increase according to antigenic strength. So this observation very much fits with the work from Anna O'Gara's lab in Mill Hill, where she's shown that in CD4 T-cells, the levels of IL-10 you get are directly proportional to antigenic strength. You can generate suppressor cells in vitro and show that the more potently you induce these cells, the more potently you suppress disease. Now, what I'd like to show you in this experiment is the logic behind dose escalation. If you go in with the highest acceptable dose of peptide, you get secretion of cytokines, and these cytokines at higher levels can be dangerous. Note this is a transgenic mouse, so these are not issues you see in non-transgenic individuals. However, importantly, this rapid cytokine production here can be prevented by starting out at a lower dose and escalating up to even a higher dose of antigen. And with the dose escalation, you don't see this burst of gamma or 2 or IL-6, and yet the IL-10 still goes up towards the end. Furthermore, following the dose escalation, these cells become relatively resistant to expression of activation markers. So, as I said, escalating up to a higher dose gives you more suppression, and you see a higher proportion of IL-10 positive cells. And the most important thing is that when you get up to these higher levels of antigen dosing, these cells are able to suppress other naive T-cells in vivo following transfer of, I think this was cell trace violet labeled cells, into a peptide treated animal. So there's no doubt that these IL-10 producing cells are capable of suppression. As I said, they're also capable of bystander suppression. And we really wanted to understand what mechanism this dose escalation protocol was inducing to bring about this change. So we're very interested in the immune synapse in my lab, and we started studying the immune synapse to see whether this in itself was disrupted in these cells. And as you'll see, comparing Th1 cells and cells from tolerized animals, you see normal accumulation of the T-cell receptor at the immunological synapse, and also normal accumulation of CD28 proteins at the immunological synapse. However, this is not true of all proteins. So proteins like ZAP70, LAT, PKC theta, they move in transiently, but then they disappear. So what's happening in these cells following desensitization is we're uncoupling the machinery required for a T cell to respond normally. Okay? This is the fundamental mechanism of this desensitization. The result of this is that the requisite signaling proteins like ZAP70 and PKC theta don't become phosphorylated as you would expect to see. So what might be causing this? What might be bringing about this, if you like, destabilized immunological synapse? Well, if you don't know what to do, you do an array, don't you? If you don't know what to look for, you do an array. And what this map here represents is a mathematical analysis of all the data arising from this array experiment. And what guys in the maths department or the computing science department in Bristol were able to do was to take the self-organizing map method to generate these clusters. And what the clusters of genes are mapped together on the basis of is the rate of change of their expression during the course of tolerance induction. Okay? Those that are increasing, those that are decreasing. And when you then actually interrogate these clusters, the clusters actually make a great deal of sense. So the genes that are coming down are those associated with inflammatory disease. Okay? The genes that are going up cluster into groups of immunoregulatory genes. Those that are going up and down are genes associated with cell division, mitosis. And then you interrogate those more closely, and you find some very, very interesting things. So if you look at the secreted factors, those that are most closely upregulated are interleukin 10 and 21. Interestingly, although the gene for IL-21 is made, there is no protein. However, there certainly is protein in the case of IL-10. Here we have a very interesting group of proteins. So LAG3, TIGIT, HAVCR2 is TIM3. And those of you in the cancer field will know very well that these are the checkpoint inhibitors. So what's happening as a result of this chronic exposure to peptide antigen in these self-reactive cells is we're upregulating expression of checkpoint inhibitors. And I think it's very interesting that some of the studies of checkpoint inhibitor antibodies are now throwing up examples of autoimmunity as a consequence of that treatment. So I think we have a balance here between regulating self-reactive cells and promoting, if you like, quasi-self-reactive cells to deal with cancer. Also upregulated are transcription factors c-MAF, NFL3, and the aryl hydrocarbon receptor, all of which have been shown by others to regulate IL-10 transcription. Now, here we're looking at an IL-10 reporter mouse, and I think it's interesting here that if you look at these inhibitory receptors, some of them associate with the anergic state of the cells. All of the cells, whether or not they make IL-10, are anergic, and most of them express LAG3 and PD-1. TIGIT and TIM3, on the other hand, associate very closely with IL-10 production. Now, it could be possible that if we bring all this together, that the disruption of the immune synapse is being mediated by high levels of expression of these checkpoint, so-called checkpoint inhibitors or inhibitory receptors. If the inhibitory receptors are drawing in phosphatases and other mechanisms, which we know happens for PD-1, it's not so clear for the others. It's likely that they would be disrupting effective immune synapse formation. So that's our current working hypothesis. So, to summarize the mechanistic studies then, effective tolerance induction may require high doses of antigen, but it appears that this can be given safely following dose escalation. And this results, dose escalation in individual cells, this results in incremental changes in gene transcription correlating with expression of genes promoting IL-10 and expression of the co-inhibitory molecules. And of course, we're hoping to use these co-inhibitory molecules as surrogate markers for monitoring tolerance induction in our clinical trials. So, to finish, I'd like to focus in now on some of the work that we've been doing to develop this clinically, and start out with an example of something that probably isn't autoimmune in nature, namely haemophilia A. So why is a guy interested in autoimmunity working on haemophilia? Well, it's location. When I was in Cambridge, my lab was just around the corner from the haemophilia centre, and occasionally you bump into people interested in other things. And the haematologists would say, look, all this peptide immunotherapy stuff is great, it's fascinating. Would it work in haemophilia? And I said, well, why would you need it? And as Ethan was talking about yesterday, in haemophilia, the biggest fear for treated patients is the development of inhibitors, of effectively neutralizing antibodies. And at the moment, very, very high doses of factor VIII are administered, but this doesn't always work, and it's incredibly expensive. So what we have done is to identify peptides from factor 8, and these are pan-DR binding peptides, 2 of which are combined in a mixture called peptide 1 and 17, F8117. And when you do a dose escalation administration of these peptides, followed by immunization with factor 8, you see a very, very substantial suppression of the T-cell response to the factor 8. So it appears that, as expected and as predicted from our work in autoimmunity, that the guys at Epitope can reproduce this with peptides against what is in effect, in these haemophiliac mice, a foreign antigen. Okay? Now, this is an experiment where we do and try to reproduce what would happen in a patient on first encounter with factor 8. And this is that after about 4 injections of protein, they start to develop antibodies against the protein. And if you do a dose escalation with peptide prior to the first dose of factor 8, then you substantially reduce the inhibitor antibodies, which are measured in this clotting assay and read out as Bethesda-like units. So it appears that we can inhibit the generation of antibodies against factor 8. And the truth is, if you come in an individual already making antibodies, you can then reduce the level of the neutralizing antibodies as well. Another disease we're interested in is Graves' disease, another antibody-mediated disease, and primarily because, of course, we know what the antigen here is. This is the thyroid-stimulating hormone receptor. Cross-linking or antibody binding to the thyroid-stimulating hormone receptor will induce or mimic TSH binding to the receptor and overproduction of thyroxine. And most seriously, there is a clear unmet medical need in terms of Graves' eye disease, which is very poorly treated at present. So again, we do a dose escalation, immunize with protein, and we can see that the T-cell response to the protein is blunted. And when you test this in Basil Rappaport's adenovirus expressing TSH receptor model of antibody production, you can see that the treatment with peptide severely blunts the generation of anti-TSHR antibodies. So this is promising. We hope that these peptides will go into clinical trials in the very near future. The most advanced clinical program that we have is in multiple sclerosis, and as you know, there are a number of different antigens that have been invoked in multiple sclerosis, including myelin basic protein, oligodendrocyte glycoprotein that I mentioned before, and proteolipid protein. Okay? Now we started out our work, we've designed epitopes from all of these proteins, but we started out our work in myelin basic protein, and we identified a range of different T-cell epitopes across the protein. But if you then look at hundreds of patients, you get a consensus that these 4 are actually the dominant T-cell epitopes. Okay? So we set about looking for an experimental model to test this in, and we took a model developed by Lars Fugger in Oxford in the UK. Actually, it was developed when he was still in Denmark, and this model expresses HLA-DR2 molecules, but also expresses T-cell receptors from a patient with MS. And what we were doing here was inducing autoimmune encephalomyelitis and waiting until the first signs of disease had already developed, and then started treating with peptide. And we're giving the peptide twice a week here, every third or fourth day. And basically, this is showing that the higher the dose you give, the more effective you control disease. And this is within the same range or the same ballpark of the top dose that we're using in the clinic. Here we're comparing the treatment with MS1467 with glatiramer acetate copaxone, which is currently one of the two leading treatments for MS. In our particular model, GA was without significant effect, whereas MS1467 was highly effective. So this brought us on to the clinical trials, and here we've taken HLA-DR2 positive individuals. There were 43 patients enrolled, and they were divided into two groups. One was given peptide intradermally, the other subcutaneously, and we're giving a dose escalation here with repeated doses of 800 micrograms of peptide, and these peptides are being given every fortnight within a few days. And the major measure that I'm going to be showing you here is the effect on MRI. Now, what I'm showing you here is just a series of images, and this is from a single patient, not in this study, I should add. And basically, this is just to emphasize to you that, in fact, in the early phases of relapsing remitting MS, this is a very dynamic disease, and these gadolinium-enhanced lesions are appearing each month in different places. Okay, so this is a very dynamic disease, and the logic is that if we could intervene very early on in the disease and prevent these new inflammatory lesions taking place, then this would be the most effective way of controlling the disease. This is true, of course, of any anti-inflammatory treatment being tested in MS. What I'm showing you here are the results from the intradermal arm of this trial. And what you're seeing here is the background level of new enhancing lesions, existing and new enhancing lesions in these patients. And then we're doing a dose escalation from here, and at this point we reach the higher dose where we're giving 800 micrograms once, twice, 3 times, 4 times, 5 times, and then stopping the treatment. We stop the treatment here, and we seem to have some longevity of the effect for about a month. Then after 3 months the response comes back. And this fits actually very nicely with data, or very convincingly with data from a previous trial where we measured the immune response to myelin-basic protein in these patients. And we found a reduction of the response to MBP at this point here, which had come back within about 3 months. So, if we look at this profile, the argument would have to be that this is not true tolerance. What we're seeing here is a dampening of activity, and presumably in order to maintain this effect we would need to continue to give the peptide antigens from there on in. Frankly, at this point I would feel quite comfortable with that, because as far as we know so far, the safety data from the 49 patients we've treated hasn't shown any unexpected concerns. So you would argue that you could keep on giving this injection of peptides continuously. So to finish, it appears then that proteins can be replaced with peptides representing T-cell epitopes for effective desensitization in our models of autoimmunity, and similar things have been found by our colleagues at Circassia who have been doing the same approach in allergy. The use of the peptides has given us an insight into the molecular changes associated with desensitization, and I've shown you the progress we're making in the clinic with these approaches. So to finish, I'd like to thank all the people in my lab who have done a great deal of the work on the molecular basis of this approach, the guys in the computational genetics group who analysed our array data for us, colleagues at Epitope and Merck Serono who are running the current clinical trials in MS, and the people at Epitope who looked at factor VIII and Graves' disease. Thank you very much.
Any questions?
Hi. Can you comment on the timing of your dose escalation? Will 4 doses close together versus 4 doses spaced out? Do you achieve the same effect?
Well, we believe that if T-cells encounter antigen very, very rapidly, they may undergo activation and induced cell death. So we believe that the most effective way of administering this is to give the peptide, have the cells activated, have them rest down, and then treat them again. Okay?
Regarding the mechanism of this IL-10 positive, IL-10 secreting T-cells, do they have you seen them proliferating also? Because they seem to be independent of IL-2, so I was wondering.
Yeah, the cells are anergic. Actually, they're profoundly anergic.
Okay.
So you know, you can grow them in vitro, and they're actually, from the mouse at least, they're very stable in vitro. But you do require IL-2 to keep them growing.
Yeah.
Okay.
Do you really think that the production of IL-10 is independent of ZAP70 PKC failure and lack?
No. It's very transient though, Ethan. So if you actually do, you know, that image that I showed you was 10 minutes, right? So in the Th1 cell, the ZAP70 lat and PKC theta are there. But in these IL-10 cells, it's gone away. However, if you look within 30 seconds, it's there.
It's not fundamentally a different signaling pathway, it's an altered kinetics of the signaling.
That's right. That's the important point. The spatiotemporal location of those signaling molecules is what's key.
So, the last time I heard you talk, I think you sort of implied that dose escalation was not required. Am I remembering that correctly?
Well, I mean, you know, in the past we've done, we've had the same effect by giving a constant high dose. However, it is interesting actually, it's very interesting, because when you do the constant high dose, you don't really see any changes in expression of FOXP3 expressing cells. Whereas when you do the dose escalation, you see an expansion of the FOXP3 expressing cells at a low dose, And I believe that they may contribute to the whole effect.
Thank you for the talk. I have two questions. One is you mentioned the dosing escalation could have effect on the efficacy. How about the route regimen? Did you try different doses, different routes?
Yeah, we tried different routes, and as many of you will know, for many years I was advocating mucosal delivery of peptides as being a very effective route. In fact, it turns out the mucosal administration of peptides is a very safe route because it doesn't work very well. Okay? Subcutaneous administration of peptide allows you to use a dose between 100 and 1,000-fold lower than mucosal administration of peptide to get the same effect.
So you think it's more tolerance, more tolerogenic than for the
subcutaneous is definitely more tolerogenic than mucosal.
Okay.
Giving peptides, that is.
And the
I think the mechanism giving proteins is going to be very different.
I see. So the second question is you mentioned the hemophilia A patient treatment, and no, the array data, and you see a lot of checkpoint inhibitory molecules upregulated, and you think it's maybe a surrogate biomarkers. But is it a little bit dangerous to have this as a biomarker, because those inhibitors could be an activation marker as well as the exhaustion markers?
Well, it turns out that, I mean, we could go back and look through the slides carefully, but as activation markers, the expression of those molecules is very transient, whereas in these cells it's more stable. So, and I think you would look, sometime after dosing with peptide in vivo to look for those markers.
Thank you.
Yes, perhaps a little bit of an unfair question to Keith, who's not here today, but how do you reconcile the data, in comparison to the data on sort of cancer vaccination, where people are trying to activate T-cells with peptide-based vaccines? Sometimes there always seems to be this question of, do those peptide vaccines potentially also induce tolerance rather than, an effective response? Does it have anything to do with the dosing?
Well, we believe, Nick, that the secret of peptide immunotherapy in our case is the fact that these peptides, given in solution, very highly soluble form, they flood the body very, very rapidly, and they bind to antigen-presenting cells, which in a resting state, namely CD11C dendritic cells in the mouse, which in their resting state are tolerogenic. Okay? And of course, some of the work that you referred to was, in the old days, people were trying to make dendritic cell vaccines, found that in fact, coating immature dendritic cells with peptide and injecting those would induce exactly the IL-10 producing cells that we're talking about here. Whereas if you matured those dendritic cells, they induce an effective immune response. So I think, and the other point about this is that if you look at those dendritic cells, both in mouse and man, the unifying feature of those dendritic cells is that they have empty MHC at the cell surface. So we think what we're doing when we're injecting these soluble peptides is having them picked up by the dendritic cells that have empty MHC at their cell surface. And just by chance, fortunately for us, those antigen-presenting cells are in their native state tolerogenic.
And do you think this is perhaps not true in the cancer setting, that those species that pick up the native peptide vaccine might not be tolerogenic?
Oh, no, but then the guys who are doing the peptide vaccination in cancer are administering other things to activate the dendritic cells, aren't they? Or they're taking the dendritic cells out, activating them in vitro and putting them back in. So dendritic cell vaccination for cancer really works, but those are activating dendritic cells, not the tolerogenic cells that I'm talking about. Okay?
Do you think, since the IL-10 is the key player here, if we give it directly with the new technology like mRNA or anything, you get the suppression of IL-17 and give you the feedback to induce this induction? So
Yeah.
the shortness.
Somewhere. Maybe it's not here. I have a slide. It's not here. And the truth is that many years ago, people tried injecting, actually at the NIH, I think it was, they did these experiments, injecting IL-10 in autoimmune encephalitis models, and it had absolutely no effect at all. So systemic administration of IL-10 isn't effective, right? So you can't just do that. However, what was effective was people taking encephalogenic T-cells and transferring into those T-cells IL-10 under the control of the IL-2 promoter, right? And the IL-10 produced by those cells, went to the site of inflammation and dampened down the inflammation. I have to say I'm a great believer in IL-10.
You've published about that in the past. I wonder what is your view now of the peptide stability and how it influences your tolerance mechanism?
Well, the evidence we have from the MBP model that we worked on, because in this MBP model, the dominant epitope is the N-terminal peptide of MBP. And this peptide actually binds extremely weakly to MHC, as do many other self-epitopes, interestingly. And you can then modify the peptide to get it to bind better. And the stronger the peptide binding, the better it works. Okay? And this goes along with that concept developed by O'Gara and others, implying that signal strength is directly proportional to the amount of IL-10 you see. So the more potently you stimulate the cells, the more IL-10 you get.
There's some reports in literature that suggest administering peptides in an emulsion, oil-based emulsions, actually promotes development of tolerance. Have you considered this form of delivery for peptides?
Many years ago, we compared giving peptide in incomplete Florence adjuvant with soluble peptide, and frankly, the peptide in incomplete Florence adjuvant proved to be activating. So we avoid that. Yeah.
One last question.
So given what is known about the mechanism, do you know if this also will work to induce CD T-cell response that's tolerizing?
Sorry, can it
be used to induce tolerizing CD T-cell response?
CD8. Well, other people have tried. I mean, Zinkenagel's group many, many years ago showed that peptide administration would ameliorate CD8 immune pathology, and I think it was LCMV infections. Hans-Peter Percher brings about. I think that, I'm showing my age now, that was probably in the early 90s. So yes, the answer is peptides have been used to downregulate CD8 responses. Whether it's working the same, at this point we don't know. And CD8 cells can make IL-10, so there's no reason why it shouldn't be.